All right, so nitrogen management. Yesterday in the discussion, I described briefly the new things that we've been learning over the last half a dozen years on the rhizophagy cycle, and what does it mean for us? How should we think about managing nitrogen differently? So there is a lot to unpack in this topic, and translating this into practical application. But the reality is that we are really doing ourselves a disservice as farmers and as agronomists by talking about nitrogen. Plants don't utilize nitrogen. Plants have zero interaction with nitrogen. Instead, they interact with nitrates and ammonium and urea and other forms of organic nitrogen, amino sugars, amino acids, peptides, proteins, etc. And this is an important point because every one of those particular compounds produces very different physiological responses inside the plant. Plants respond completely differently to nitrate than they do to ammonium or urea. And from an overall plant health and yield perspective, these differences become very important. So I believe that we would be well served to not continue to have conversations about nitrogen because plants don't interact with nitrogen. So when we think about building stable soil aggregation and building soil carbon levels at a significant depth, when we have the presence of free soil nitrogen, shutting down the rhizophagy cycle, shutting down the soil microbiome, this is one of the most effective applications or one of the most effective means of preventing the rhizophagy cycle and preventing fungal endophytes, bacterial endophytes from colonizing the plant and from forming an effective relationship with the remainder of the soil microbiome and building overall soil health. So I attempted to locate a diagram that describes the nitrogen cycle as thoroughly and as completely as possible as we understand it today. I was able to find this diagram. But you know what? Even this diagram does not, this diagram still does not consider the contributions of soil biological nitrogen to the crop itself. So therefore, it's still incomplete. This is thinking about nitrogen largely in terms of mineral ionic nitrogen or inorganic nitrogen sources being contributed to the plant. When we think about how many of you did not listen to me describe the rhizophagy cycle in my presentation yesterday morning? Okay, wow, most of you did not. All right, so when you keep giving the same presentation 100 times over, you think everyone has heard it after a while. All right, so this piece of information is beautiful and awe-inspiring. Almost 2 decades ago, I realized that some of what we were observing with plant health and plant growth, I had observed trees growing out of hard rock faces where there was no topsoil present, and it became very clear that this model of plants absorbing simple ions from the soil solution could not be the mechanism and the model that was supplying nutrition to plants in undomesticated ecosystems. And I think it's important to back up for context so that we realize the implications. So there are 2 models of plant nutrition that are widely considered today. The first model, and the model that is used in all of mainstream agronomy and horticulture and plant nutrition, is the model that is based on the idea that plants have the ability to absorb simple ions from the soil solution, from the water solution, whether that be calcium, magnesium, potassium, sodium, ammonium, nitrate ions from the soil solution, that they then absorb those ions, transport them throughout the entire plant, and that those ions are then utilized as a source of nutrition by various plant cells. And it's true that plants have the ability to do this. After all, this is how they function when they're in hydroponic ecosystems. But there is a second model which has recently been modernized by Dr. James White and his colleagues and called the rhizophagy cycle. And this model is based on the idea that plants can derive their nutrition from living microbes. And in his work and his colleagues' work, they have identified that plants are colonized intensely by bacteria and fungi that are due to living inside plants. These are called associated endophytes. And endophytes are simply an expression of the fact that these bacteria and fungi live throughout the entire plant vascular tissue. They are in the seeds, they are in the fruit, they are in the grain, they're on the plant leaf surface, in which cases they're often referred to as epiphytes instead of endophytes. They live throughout the plant's entire vascular tissue and even inside the cells. So in this model, we have plants deriving nutrition from microbes rather than from simple ions from the soil solution. And the question is, how much of a crop's total nitrogen supply and nitrogen requirement can be contributed with this process? And not just nitrogen, but with nutrition overall. And I'm persuaded that the answer is upwards of 90%, 100% for many different crops. So let's talk a little bit about the rhizophagy cycle. What Dr. White and colleagues described is that the growing root tip, as it is growing through the soil profile, is actually very porous, and that there are large volumes, large quantities of bacteria that move into the root tip, and after they have been absorbed into the root, there is a highly oxidized zone in the first couple of inches immediately behind the root tip. This highly oxidized zone, plants release various oxidizing species, particularly nitrous oxide, and strip the cell membrane off many of the bacterial cells. And then these naked bacterial cells now move through the plant's vascular tissue and are transported throughout the entire plant and are utilized by the plant cells as a source of nutrition. But in this process, when bacteria move back from the growing root tip, a few inches back from the growing root tip, some of these bacteria begin to accumulate. And as they begin accumulating, they trigger the formation of root hair. As the root hairs lengthen, these naked bacterial cells now migrate out through the root hair and back into the soil environment. And the plant, as it releases these bacteria back into the soil environment, it also releases sugars and mucilage, mucigel compounds that facilitate the microbes being able to quickly reform their cell membranes. Then, in this process, as microbes are being absorbed by the root hairs or root tips, moving back through the root system and back out into the soil system, the plant is also communicating with these microorganisms and is, through both chemical signaling and electromagnetic signaling, is communicating to them exactly what its nutritional requirements are. It is communicating to the microorganisms that I need more phosphorus, or I need more manganese, or I need more zinc. And then as these bacteria go out through the root hair into the bulk soil environment or into the rhizosphere, they then communicate and share those signals with the rest of the soil microbial community. And the rest of the soil microbial community will rapidly begin extracting these particular minerals that the plant requires out of the soil mineral matrix. So they will begin selectively increasing, absorbing larger levels of zinc or manganese or phosphorus. And then those bacteria that are now loaded with higher concentrations of these minerals are then selectively picked up by the mycorrhizal fungi network and channeled back into the plant. And now the plant gets nutrients from these living microbial cells. Isn't this a beautiful process? This is a completely different understanding and a completely different framework of understanding plant nutrition. And the reason this is so important is that understanding this process and its degree of effectiveness completely invalidates every single agronomy and plant nutrition textbook and paper that has been written in the last 70 years or longer. Any article that you read on or a peer-reviewed paper or books that are published on plant nutrition always have the obligatory opening paragraphs or opening chapters on how plants absorb and transport ions, but no mention of how plants absorb and transport nutrition in the form of bacterial cells. So this entire process, the question that I asked a moment ago is how much of the crop's total nutritional requirements and nitrogen requirements can this process provide? And I started asking this question roughly 7 or 8 years ago. We had an experience with a farm that we were working with in south central Kansas. So this is a certified organic farm, dry land, no irrigation, roughly 15 to 17 inches of average annual rainfall. And 20-some years previously, this soil had a history of significant livestock manure applications for about an 8-year period, and then nothing for the following 20 years. Because of the dryland conditions, the prevailing belief was that to have effective weed control with an organic management system, there was not enough moisture, not enough rainfall to have a cover crop. So there was no cover crop being grown. It was simply corn on corn on corn, and dependent on moisture conditions, they would occasionally only grow corn in every alternate growing season. So that meant that there was no nitrogen coming into the system. There was no compost being applied. There was no manure being applied. There were no cover crops being grown. And yet, when moisture levels were adequate, this farm was producing 220 to 250 bushels per acre of corn, completely organically. No liquid fish is being applied. There are no significant sources of nitrogen coming into the system. And this didn't occur just 1 year or 2 years, but we worked with this farm for several years, and we observed, we're of course using microbial inoculants that include azotobacter and azospirillum and other nitrogen-fixing organisms, but it became clear that there was something happening in this ecosystem where there was a great deal of nitrogen coming into the system that was not being accounted for through the typical delivery mechanisms that we commonly associate with being needed for organic nitrogen production. And flash forward to more recent, the last couple of years, we have Walter Goldstein from the Mondawmin Institute, who's someone I want to host on the podcast soon, has described, he has been selecting corn varieties that are, I'm not sure what the correct terminology is here, not ancient genetics, but that are older or original genetics coming from Mexico, and he has been growing these old selections of corn cultivars in specifically in nitrogen-deprived environments, growing them where with no manure applications, no cover crops, no compost, no synthetic manure, or no synthetic nitrogen of any type. And he's been selecting varieties that are producing abundant levels of nitrogen, all that the crop can require. He started collaborating with Dr. White from Rutgers University several years ago, and they were able to identify that, without question, his corn selections are associated with nitrogen-fixing bacteria, nitrogen-fixing endophytes that are embedded into the corn seed. They're propagated from generation to generation, and they colonize the entire corn plant, and they're capable of completely delivering 100% of that corn crop's nitrogen requirement. Isn't that incredible? But they also have identified that if you want to shut that system down, it's very easy to do so. You simply apply synthetic nitrogen, and it's game over. The associated microbiome with the microbiome associated with that corn plant will not continue to fix nitrogen for the remainder of the growing season if synthetic nitrogen is applied to that crop. So the piece that we need to begin thinking about is, how do we begin managing our agronomic systems so that we avoid excessive soluble nitrogen in the plant and in the soil, because this is the foundational piece that leads to so much disease susceptibility and insect susceptibility, as I described in my presentation a little bit ago. So, of course, in order to have reasonable nitrogen fixation in the soil process and with the microbiome that is inside the plant and in the rhizosphere, we need to have good gas exchange. We need to have good airflow into the soil profile. Now, here's another piece of the puzzle, which is that in our experience, when we are harvesting reproductive crops, such as fruits, vegetables, grain crops, etc., very frequently the highest yielding crops have less vegetation rather than more. Why is this? I'm not sure if I've included it in this slide deck or not, but we've observed that plants that absorb nitrate versus ammonium produce completely different vegetative and reproductive balance responses. So what I can say generally, I wrote a blog post on a related topic a couple of weeks ago where I described that rather than overall crop canopy, there is this paradigm of associating overall leaf index, overall crop canopy with yield capacity, the capacity to carry fruit and support fruit. But in reality, there is a much more direct comparison between individual leaf size of specific fruit-associated leaves rather than overall plant canopy. So this is actually, I don't recall exactly which stage this photo was taken, but this is a photo of a record-yielding watermelon crop that we harvested over 70 tons per acre from. And at harvest, the plants did not completely cover the plastic rows. They had put much more energy into the plant, into the fruit than they had put into plant vegetation. And this is an expression of we have become acclimated because nitrogen applications have been easy. We have become acclimated to the idea that we need to have lots of vegetation. We need to have a lot of vegetative biomass, and that is the foundation for producing high-yielding crops. And so we tend to overuse nitrogen as a result. A year or two ago, I conducted a webinar that is still available on YouTube of how nitrogen creates yield drag. And here's a quick overview. It results in excessive vegetation. It increases the plant's water requirements. It drains plant energy. It reduces nutrient absorption of other nutrients because it creates an alkali rhizosphere environment. It increases disease and leads to premature death. So let's give you a quick synopsis of each of those materials. First, how does nitrate fertilization increase a plant's water requirement? In the nitrate conversion process when nitrates are absorbed to convert nitrate to glutamate and to amino nitrogen, requires 3 molecules of water for every molecule of nitrate. So if you want to increase a plant's drought susceptibility, it's really easy. Add more nitrate. What happens in the rhizosphere when plants absorb nitrate versus ammonium? These two elements constitute a great deal of the cation charge, the total charge distribution of what is taken up by the plant, and therefore has a strong impact on the other cations and the other anions that plants will absorb, and on cellular pH regulation, rhizosphere pH, and so forth. So here's a key point. Absorption of ammonium acidifies rhizosphere pH and generally leads to better absorption of other plant nutrients, better absorption of manganese and zinc and iron and copper and phosphorus, iron and copper, etc. But the absorption of nitrate has the opposite effect. It alkalizes rhizosphere pH and results in reducing the absorption of many of these other trace minerals. So, and this is particularly true of calcium and magnesium and the trace minerals. So, in addition, nitrate also produces weak and watery cell membranes that are much more susceptible to disease and insect attack, bacterial and fungal infections, etc. We have found, we work with many fruit and vegetable crops that are stored for long periods of time, apples, onions, potatoes, etc. And as well as fruit that need to be shipped and they need to hold well, cherries, strawberries, extensive list. Greater than 80% of fruit quality problems are a direct result of the excessive applications of nitrogen and potassium. If we were to simply avoid the excess applications of those two elements, we would resolve most fruit quality problems very quickly in a very straightforward manner. In my presentation an hour ago, I described the energy requirement needed. I'm not following the slide here, but when plants absorb nitrogen, it costs them a lot of photosynthetic energy to convert that nitrate to complete protein. So overall, this is just observational experiential data distributed across a lot of farms. We have not published this. When plants absorb the majority of their nitrogen in the form of nitrate, it seems to increase the plant's water requirements and increase the evapotranspiration rate. In order to maintain plant turgor and good photosynthesis, it increases water requirements by roughly 20 to 30%, depending on the plant species and context that we're talking about. And of course, you can greatly reduce this water requirement simply by utilizing nitrogen from the rhizophagy cycle. This is research that was conducted at UC Davis. This is a summary paper that was handed out at one of our events of the efficiency and the effectiveness of foliar-applied versus soil-applied nutrients. And I included this because of the nitrogen number. Their research indicated that 1 pound of foliar-applied nitrogen was the equivalent of 7 pounds of nitrogen applied to the soil. That's a pretty significant bump. You want to reduce your nitrogen fertilizer bill by 80+%? Simply begin foliar applying rather than soil applying. I'll go back to that. See lots of cameras in the air. I should have included the citation. The citation got cropped off in the photo when I blew it up. All right. Nitrate also creates yield drag by increasing excessive vegetative growth. It has a synergistic relationship with the plant hormone auxin, and leads to wide node spacing, less reproduction, increases water requirements, increases energy needed for nitrate conversion, etc. We reviewed this. All right. So, what we have learned from practical application is that if you want to, actually let me back up just a step. Our approach, given what we have learned over the last couple of years, how do we manage nitrogen today versus how we used to manage it 20 years ago? And there are two pieces. First, we use microbial inoculants that contain species that are known to sequester and fix nitrogen given the right environment. Actually, I'm going to mention this other piece as well. We live in a world today, there are dozens of products available in the marketplace, hundreds in fact, but dozens in particular who claim to have the ability to fix nitrogen and make nitrogen available to the plant. At least in the North American marketplace, these products have greatly proliferated. And farmers apply them. Sometimes they are foliar applied, sometimes they are soil applied. Some of these products require multiple applications per season. And there was some research conducted 2 years ago. I think the report was released in Canada where they evaluated 62 crop years of these microbial inoculant applications, and they observed positive nitrogen contribution in 4 out of the 62 crop years. That's a pretty dismal rate of success. But that's because they added microbial inoculants into the planter solution with liquid nitrogen. It's like, how obvious can you be? It's remarkable that they got 4 successful crop years out of the 62. So our approach is that we use microbial inoculants and biostimulants as seed treatments and in the furrow at planting with no nitrogen application at planting, unless we know for certain that the soil and the plants, or that the soil is extremely nitrogen deprived and that we may need to add a small amount. So we add no nitrogen at the beginning because the reality is that a small seedling in a small corn plant or rape or wheat plant, the nitrogen requirements are extremely low. For many of these crops, the majority of the nitrogen requirement, upwards of 70%, in many cases upwards of 80%, doesn't occur until the grain fill stage. We need nitrogen late. We do not need it early. And so the key is to establish this microbial population early in association with the root system and to not suppress it and not shut it down. So one of the very simple pathways, in addition to there's two pathways here, two thoughts. One is that, yes, you add the microbial inoculants, you add the biostimulants, but then part two is that if you want those microbial inoculants to succeed, you have to discontinue the nitrogen application up front. You cannot have nitrogen applications in close proximity to the microbial inoculant and expect the inoculant to be effective. It's simply not going to work. So our approach is we put on no nitrogen applications up front, and I'm focusing this conversation on nitrogen, but the reality is we also do the same for potassium. No potassium applications up front. And then we use sap analysis through the growing season at critical stages of growth to evaluate what the crop's actual nitrogen requirements are, and then to spoon feed nitrogen through the growing season as needed with foliar applications. And also let me just clarify. To spoon feed with foliar applications are also to use side dress applications. So we use both foliar applications and side dress, depending on the farmer context, what equipment, machinery, and capabilities that they have available. So with foliar applications, remember the rule of thumb that I showed you just a bit ago is that 1 pound of foliar applied nitrogen is the equivalent of 7 pounds of soil applied nitrogen. So there are tremendous efficiency gains from foliar applications. And the key also is that foliar applications do not have this effect of shutting down the soil microbiology the same as soil applications do. But when we need to make side dress applications, where did it go? Here it is. Let me see what I have here. All right, let's talk a little bit about which forms of nitrogen to apply and how to manage them. So when we consider the plant's nitrogen utilization efficiency, we have four different forms of nitrogen. The most efficient form is listed here as amino sugars, but it should actually say organic nitrogen, because organic nitrogen includes amino acids, amino sugars, peptides, proteins, etc., and bacterial cells. The second most efficient form of nitrogen is urea, and urea is one of the most common forms that we apply in non-organic systems as foliar applications. The third most efficient form is ammonium, and the fourth is nitrate. Nitrate is the least efficient and costs the plant the most energy. You know, it's quite interesting. We think of nitrogen as giving plants energy, right? But yet, nitrogen, we come to find out, actually creates yield drag. So as we are in the process of transitioning a soil and cropping ecosystem to reduce the nitrogen requirements, it is critical that any nitrogen that is applied to the soil, and this conversation is specific to soil context, that any applied nitrogen is converted to organic nitrogen, to bacterial nitrogen as quickly as possible. If we are in a situation where we need to add nitrogen to the soil, whether that's as ammonium, UAN, nitrogen, urea, stabilized urea in any form, we want that nitrogen to quickly be converted to bacterial cell protein nitrogen. We want it incorporated into living bacterial bodies. And this gets us to a beautiful place. William Albrecht said decades ago that plant nutrients should be available but not soluble. And this is facilitated when we have bacterial proteins as our form of nitrogen in the soil profile, because they are very readily available to the plant, but they are not soluble, which means that they do not leach and that they remain available in very dry environments. So our approach to stabilizing nitrogen when we need to add nitrogen to the soil profile and to facilitate rapid bacterial mineralization, is to our nitrogen solution we add soluble carbon, sulfur, and molybdenum. And here's what it looks like. Actually, this slide needs updated. But we look at, when we consider carbon to nitrogen ratio in soils, stable organic matter, nitrogen to sulfur ratio, you can combine these to look at this overall common denominator of 200 parts carbon, 10 parts nitrogen, 1 part sulfur. And you could argue that this depends a little bit on the lab methodology that is used to measure carbon. You could also equally argue for 500 parts carbon to 10 nitrogen to 1 sulfur. But here's the key part. Nitrogen in the nitrification process, nitrogen will convert to nitrate in the soil profile based on the lowest common denominator in this ratio. And frequently the lowest common denominator is sulfur. So when we put together a solution, we will add sulfur to achieve a 10-to-1 nitrogen to sulfur ratio in the mix. So we're commonly adding ammonium thiosulfate. If you're using urea, you can utilize ammonium sulfate. but we add sulfur to the combination to get us a 10-to-1 ratio. And this provides enough sulfur to form all of the stop amino acids, the sulfur-bearing amino acids, to form complete proteins inside microbial cells and inside plants. Then we add humic acid. So these are humic substances. Well, it's technically not. This humicarb is not a humic acid. It is technically it's a mechanically extracted humic substance, and we utilize it at 3% of the total nitrogen solution. And again, here I'm referring to liquid solutions. This can also be utilized dry. But 3% of the solution, if you're using humic acid, you will need roughly double this, between 5 and 6%. And this is enough to complex and stabilize a great deal of the nitrogen that is in the solution and provides a substrate for rapid mineralization and rapid incorporation into bacterial cells. And then we add a pint of, I think this product is 6% molybdenum, if I recall correctly. 1 pint per acre of molybdenum into our nitrogen applications, being the enzyme cofactor for the nitrate reductase enzyme. And lastly, we add this material, which is partially soluble carbohydrates, to the total solution volume. So this is what the total combination looks like. And we find that when we add this combination to soil, instead of, let's just say, liquid urea, or we have measured, when we add this to a liquid sidedress application, it reduces, actually, I think I might be missing those slides. Oh, there it is. Let's go back. Okay, I've already focused on these pieces. So that combination results in, or of not using that combination results in 16 times more nitrogen leaching out from the soil profile as nitrate. In other words, when we have added that combination of sulfur, humic substances, soluble carbon, and molybdenum, that facilitates very rapid conversion of the applied nitrogen into bacterial proteins, into bacterial cells, and it remains plant available but not soluble. So, there are pieces that I missed. I do apologize. I'm realizing I'm still a little bit jet lagged, and I've had 2 intense days of nonstop conversations, and so I've had tons of fun. But my brain focus is no longer where it was even for the conversation earlier. So I'm sure there are pieces that I've missed, but not sure how we're doing on time, but we're going to open it up for Q&A. We have a roving microphone in the back if anyone has any questions. My presentation mentally didn't feel quite as coherent this afternoon, so my apologies for that. Yeah. Thank you. Thank you.
I have a question about the rhizophagy cycle. And there is these multiple schools of nutrient uptake by plants. And we see that the traditional mineral inorganic-based pathway is the least effective. And then it was a huge revelation for me when I learned through the Soil Food Web School that microorganisms who live in the rhizosphere are eaten by predatory microorganisms, and they are putting out their manure right in the rhizosphere for the plants for uptake. And now you're saying that the rhizophagy cycle is responsible for pretty much 100% of the nutrient uptake. So I'm really curious about your opinion about these microscopic manuring of the plants. And yeah.
Yeah, so I just want to clarify, I'm not suggesting that the rhizophagy cycle is 100% responsible for plant nutrition. I'm saying it can be. It has the capacity to supply 100% of the plant's nutritional requirements. The having bacterial populations and microbial populations grazed upon by protozoa and cycling and releasing those minerals is still an important and valuable mechanism. But the reality is that we again have, rather than individual ions, we again have compounds and complex molecules that are being transported by mycorrhizal fungi or being absorbed by roots directly. So it is why in the plant health pyramid, conversation, I framed the conversation as microbial metabolites, plant nutrition from microbial metabolites. Those can be bacterial cells, or they can also be microbial necromass to some degree. Yeah. We had a few more questions up here in the front, one here and the one over here.
My question is, with companions in crops, can they, is there a synergistic sharing of nutrients via microbes or fungi when there's abundant lipids, like, would you call it the fat of the land?
I would call it exactly like the fat of the land. So I'm now remembering one of the points that I missed mentioning. When James White started researching Walter Goldstein's corn selections for the microbial populations that were so successful in fixing nitrogen, they were able to identify that each corn seed contains 9 billion microorganisms. So we need to begin thinking about seed as a microbial inoculant. Seed should be vectoring the specific microbiome that it is associated with from one generation to the next. Now, this frequently fails to happen or fails to happen well, and there's any number of reasons for why this occurs, but one of the reasons is that often we don't have a functional microbiome associated with a plant because we've compromised it with fungicide and herbicide applications and with our modern management practices. But also a great deal of it has been lost over multiple generations of high technology breeding work. And they've identified that one of the best ways that to greatly compromise and reduce the complexity and diversity of a microbial community associated with a specific plant is to propagate it through cell culture. And a great deal of propagation and selecting and breeding work happens today through cell culture. So when you have a cell culture, you are completely sterilizing it, and there is no associated transfer of the microbiome from the parent generation to the daughter generation or the child generation, and therefore this follow-on generation needs to freshly recruit microorganisms from the soil that it is eventually planted into, which frequently results in a very different microbiome from what the parent generation originally had. So you have this loss of transfer, but it seems to be possible to re-inoculate that. They have established that, at least for some varieties, it is possible to take the corn that has this associated microbiome capable of fixing nitrogen and transfer that microbiome to another variety. So the question is, will that hold true for all varieties? That's something that yet remains to be discovered. But to the fullness of your question, when there's something fascinating that happens when plants have abundant levels of lipids, when you have this microbial association to begin producing a surplus of energy, at that stage, when plants have high lipid levels, they also transfer lipids out through the root system as root exudates to feed soil biology, not just carbohydrates alone. Well, we know that it's much more than just carbohydrates. Carbohydrates are the majority, but at this stage, and particularly as plants begin approaching maturity, you also have a lot more lipids that are sent out through the root system. And this is fascinating because when you have bacteria, for the most part, cannot consume and digest and break down fats. So when you have lipids going out through the root system as root exudates, this triggers fungal digestion. And this fungal digestion of lipids is what leads to rapid formation and rapid development of stable humic substances that have a long-term shelf life in the long-term life in the soil profile. And that is the fat of the land. Yeah. Sorry, yeah.
Hi there. I was educated long ago, so it was when ions were the thing, and it's great to hear your new research. How would you apply it to grazing grassland?
It directly applies to grazing grassland, and as I understand it, to all plants. So, did I understand your question correctly? Yeah. So, if I'm understanding your question correctly, are you asking whether we are managing nitrogen in the same way? Is this in context of nitrogen? So the question is, in grassland environments, how are we managing nitrogen since we are not taking the crop all the way to maturity, but we're harvesting it while it's still in a vegetative state? Did I capture that correctly? The answer is that we are also using sap analysis and measuring the sap profile of nitrogen and using that to make informed decisions about whether the crop actually requires additional nitrogen or not. And it's been our observation that, again, it depends on the rest of the agronomic management system, but grassland is particularly effective, particularly when it is combined with broadleaves and we have a diversity of species. It is particularly effective at, the microbiome is effective at fixing and supplying all of the crop's nitrogen requirements once we stop adding synthetic nitrogen. You know, it's important for me to communicate what we are doing and the successes that we've observed and some of the actual management practices, but I also want to mention that I want to be cautious about oversimplifying it. It requires systems level thinking, systemic thinking. So as the example, the answer that I've just been providing, yes, it's true that many grasslands are capable of fixing nitrogen very effectively and very efficiently, but in order to do that, you have to address molybdenum, you have to address cobalt, you have to address the enzyme cofactors that are required by the microbiome to be able to fix nitrogen well. So I want to just emphasize that in order to achieve these levels of results, you have to take a thorough systemic approach. Evaluate the soil microbiome, evaluate the actual plant nutritional status. Don't guess about these various things and make sure to address all of them systemically so that you can be confident of success. And on that note, I almost missed mentioning this. I've been in close touch with a number of you. I've developed many close friendships here in the UK over the years. This is the first time that I've been here in person, and there have been many requests. We are considering bringing our consulting work and eventually our products here to the UK as well over the course of the coming months. And I'm curious, how many of you, if we were to establish a consulting service to share what we have learned here in the UK, how many of you would be interested in working with us in some way in that regards? Okay, wow. All right, well, see Kish over here in the corner. Yeah, there's obviously lots of homework required to bring that here and to make it work well, but Kish can succeed in pulling it off. We have another question. Okay, yes.
Do you have any foliar application recipe, or that would help for weeds if you have an immediate problem, and I mean that you make them sick or they stop growing or whatever?
Oh boy. We have had foliar applications that suppressed weeds very significantly and immediately, but there hasn't been a consistent pattern. And for example, we had one farm that the soil and the crop analysis showed that they were consistently low in calcium and manganese, and so we put together a recommendation that included calcium, it included manganese, it included some biostimulants. This was on a hay field with severe problems with Canadian thistle, and the grower put on the application. The forages responded extremely strongly, and the Canadian thistle completely shut down. And, oh, well, this is an awesome idea. Let's see if it works elsewhere. We take the exact same recipe, and we take it onto other farms, and we get mixed results. It works maybe 30% of the time. And so I believe that we happened to be lucky enough to strike the perfect combination that was a reflection of what that particular soil and that particular context needed at that moment, but that's not necessarily a perfect reflection of what other soil combinations are particularly looking for. So we have a few of those things, a few of those recipes that we have tried that have some level of success, but they're not consistent enough that we can confidently make recommendations around them. Yes.
Hi. Tracy Worcester. I'm deeply concerned about the future of food, which seems to be saying that we don't need soil. So you get your vertical farms with the injection of fertilizer, with the injection of water. And I wondered what your opinion of that sort of future is.
Very good question. My experience and observation has been that I'll slightly rephrase your question to rather than say that we have a question right here. Rather than the absence of soil, I'll frame it as the absence of microbes, because it is possible to manage some of those systems so that we have abundant microbial populations. The simple answer is that, as I described the plant health pyramid, our experience has been it's relatively easy to get to level 1 and level 2. impossible to get to level 4 without biology. So biology does less well on synthetic substrates, or I should say on artificial substrates, but it can still be functional, but it is still not the optimal results that you will get in a soil environment. So when you look at this purely from a medicinal value, phytonutrient concentration perspective, it's my opinion that growing in real soil is going to exceed the benefits and the results of growing in these artificial substrate environments.
So our health will be impacted if we grow in the water and the full.
It could be. However, the follow-up comment was, so our health will be impacted. Well, it could be, but the reality is that many crops that are grown even in topsoil today are not at the levels of health that, let me say it this way, you could grow healthier crops in the most healthy hydroponic substrates than you can in the least healthy topsoil. If that makes sense. Yeah.
John, Stafford Proctor, thank you very much for giving your heart and soul the last 2 days. We've watched all of your seminars, and they've really been amazing. Thank you for that.
Thank you.
I hope you get some chance to relax before
Next
you go
week.
back home.
Thank you.
John, my 2 questions. You mentioned about the early application of nitrogen and potassium, and then right at the end you slipped in phosphate as well. Now, I know phosphate is vital for root growth, so you've confused me a little bit on that one. And the second part of my question is neutral pH for applications of foliars, and I guess pesticides as well. How important is rainwater harvesting to your experience of that?
Okay. Both very good questions. I'm going to answer the second question first. I put together a webinar. This is such a common question that I put together a webinar on how to create a well-designed foliar spray that is consistently effective. It's available on YouTube. It's one of our very popular webinars. It's an hour worth of information. I'm not even going to try to summarize it here, but we talk about the need for clean water sources, where to balance pH, where to balance electrical conductivity, where to balance the point of deliquescence, in which sequence to add different types of nutrients. The information is all there. To your first question, phosphorus requirements at planting to establish a large root system. Well, there's this little secret, which is that when you utilize magnesium in a planter solution, you will generally succeed, at least in our experiments, you will generally succeed in producing larger, more robust root biomasses than you will when you add phosphorus. With the additional bonus that you don't shut down the mycorrhizal fungi network as you do when you add phosphorus. So if you add soluble phosphorus at planting, it is one of the most effective ways possible of shutting down the mycorrhizal fungi network and preventing it from producing a colonization. And on that note, I'll just expand on this for just a little bit because it is important. Many of the root-borne diseases, the root pathogens, the susceptibility to those pathogens is often set up in the first 14 days after germination. The disease might not show up for weeks or months later, but the initial infection or the initial susceptibility is often set up in that first 2 weeks. Because what should happen, ideally, we have a healthy microbiome inside the seed and on the seed surface. Of course, we then in our wisdom desire to destroy that and add fungicides to the seed surface. But then what happens is the seed germinates, the root radical emerges, and in a perfect world, that root emerging radical and the initially developing roots are immediately colonized by a healthy microbiome that is coming from the endophytes inside the seed as well as the microorganisms that are on the seed surface. And so that we have this root system immediately colonized both by mycorrhizal fungi, by bacteria, and have this very rich rhizosphere. In our common agricultural systems, this commonly fails to happen because we commonly apply either nitrogen or potassium or phosphorus fertilizers or some combination in close proximity to the seed, and that creates an environment with artificially high levels of these soluble nutrients. The root system begins extending and growing very rapidly, but more importantly, the plant gets the signal that it lives in a nutrient-rich environment. it doesn't need to share carbon. It doesn't need to share carbohydrates with the soil microbes. It doesn't need to develop an association with soil microbes because it has abundant nutrition. And then over a period of time, that abundant nutrition begins to fade away. And now the crop would benefit from having an associated microbiome capable of supplying nutrition. But at this point, you're weeks down the road, and you now have a large proportion of the root system that hasn't been fully colonized to the degree that would be possible. So what we need to have happen in a very healthy ecosystem, to produce very healthy crops, is we need to have complete root colonization very rapidly, immediately after the seed germinates. And what happens is that often we have a failure to completely populate and colonize the root system for a 10 to 14 day period or sometimes longer after germination. And it is in that window of time that pathogens produce their initial infection. And then those pathogens are often, there is a temporary resurgence of the microbial population. Those pathogens are temporarily suppressed. But then months later, weeks later, those pathogens first begin producing the visual expression of a disease. And we call it a disease, failing to understand that the original infestation occurred as a result of a compromised microbiome in the initial 14 days right after germination. Yeah. Yes.
That's actually really interesting leading into my question, which goes a bit back to the intercropping question. I was thinking about the three sisters and trying to use legumes to add nitrogen to other plants that need them. I'm doing that right now with corn. And I'm thinking about that kind of overabundance of nutrients leading to maybe lack of recruitment of the microbiome. Is there a certain timing that would be optimal for that sort of thing? Or, planting them exactly together, or any kind of combination, just that mechanism I'm curious about.
My understanding is that the indigenous Americans planted them at the exact same time. And it's also my understanding that you need to be less concerned about legumes producing elevated levels of nitrogen and compromising the colonization when there is abundant carbon in the soil profile. So when there is abundant carbon, then you buffer out any free nitrogen that might be supplied by the legumes, and you minimize the possibility for that happening. Yep. All right, time for one more question. Yes.
Hi there. I have a question regarding your thoughts on crop rotation. Is it really as beneficial as everybody thinks it is?
You like asking the questions that get me into trouble. I've been known to have many heretical opinions, so I might as well continue the trend. If you would have asked me that question 15 years ago, in fact, someone did ask me that question 15 years ago in a meeting in Pennsylvania, and my answer at the time was that I believed that when we have a highly functional microbiome that has produced a disease-suppressive soil, there are not significant benefits to continued crop rotations because you can actually adapt the soil microbial community to that particular crop. However, on the pathway to developing a disease-suppressive microbiome, crop rotations could be very beneficial and could be valuable in getting to that point. That was my answer to the question 15 years ago. One of the farmers in the room who heard that response was having major problems with fusarium on watermelon. And to the point where he was having significant crop losses, and this was on a farm with a 7-year crop rotation. And in that 7-year period, he was making very certain a few of those years to remove any host species for fusarium, which is a challenge. And he chose the bold decision of discontinuing his rotation while he was still having significant disease pressure. He planted watermelon on watermelon on watermelon year after year. By year 4, his fusarium pressure had dropped from 70% plant infection initially to 0. Now, my understanding today, I want to be very clear, while he was growing watermelon in the same soil every year, I think a critical piece of his success was that while he was planting watermelon every year, watermelon was not the only plant in the soil. He, in fact, had a a cover crop blend between his rows of plastic mulch that was constituted of dozens of species. And so I think it was the contribution of those dozens of species present every year that was actually contributing a great deal of the disease suppressiveness effect. So I do think that perhaps there are good reasons for crop rotation. But when we include a diversity of plant species from many diverse groups and different plant families, we can develop a disease suppressive microbiome that doesn't require the need for crop rotation purely for the purpose of disease suppression. All right. Thank you very much. I hope you found the information valuable. Thank you.